are dependent on water, right? Yeah, the amphibians are dependent on the water. And then the reptiles or the amniotes, they try to be less dependent and can go further away and to different habitats which may not really have bodies of water. And for this reason, they really have to minimize the water loss that they could be experiencing. So animals like the amniotes and basically the reptiles would have better developed skin to help with minimizing this water loss. Now, the skin is the site for synthesizing and accumulating lipids, vitamin D, and reserves of water or blood. And it acts as an insulator in birds and mammals. We mentioned that birds and mammals they belong to a certain group. Does anybody remember what group that was? Like a certain group of animals. What do birds and mammals have in common? Or okay, okay. So we have endothermic animals, and then we have exothermic or ecothermic animals. And what was the difference? Does anybody remember the difference between those two groups? No. Okay. So endothermic animals are animals which are able to regulate their own body temperature, regardless of what the external temperature is. And usually they're able to generate this heat as they're able to break down their nutrients, or they're able to basically use metabolism in order to generate that heat. Whereas the ecothermic animals, they are animals like the reptiles, the amphibians, and the fishes. They're not really able to regulate their core body temperature, and as a result, they also what's it called? They don't really rely on their metabolism in order to generate heat. They get their energy basically from the sun. Okay, so sensation. So of course the skin has got nerve endings for touch, pressure, temperature, vibrations, and pain. And our skin is able to absorb and excrete substances as and when it needs to. And in amphibians, it is an important respiratory organ, as amphibians are able to breathe through their skin. And then, of course, the skin has got some level of pigmentation to it, and it serves purposes such as mimicry, which is when animals need to attract a mate, and so on. Okay, so what is the structure now of the skin? So this is important to note also. So there are three main layers that you need to know. So we have the epidermis, which is the outermost layer, then the dermis, which is right underneath it, then the hypodermis, which is the layer that's all the way at the bottom. Now, these three layers are different in their function and in what's basically inside of them. So the epidermis, it comes from the ectodermal, which is the outermost germ layer. And then the dermis comes from the mesodermal area, which is, of course, the what's it called, the middle germ layer. And the dermis usually has fibrous connective tissue, and this connective tissue is there to basically support the epithelial cells which are above it inside of the epidermis. And then the hypodermis usually's got subcutaneous tissue where the nerves are traveling through, and it also has loose connective tissue there and adipose cells. And what is the purpose of these adipose cells, or what's the purpose of adipose tissue?
Is it to store fat?
Okay, yes. And why is that necessary?
It
Why is
helps
it
with
Sorry?
it helps with a lot of things. Insulation,
Insulation, exactly.
supports structural functions,
Mm-hmm.
even like for energy storage and metabolic roles as well.
Exactly. So The two main reasons are, as you said, insulation. So fat basically gives us that insulation, because remember, as we said, if we're talking about ourselves, if we're talking about humans, we are endothermic animals, where we're able to regulate our own body temperature using homeostasis, and the adipose tissues help us with this. The secondary role of the adipocytes within the adipose tissue is to store fat. And basically, the purpose for that is our body likes to break down glucose mostly. It likes to use carbohydrate metabolism in order to generate ATP. So we're breaking down glucose to get ATP. And that is our preferred way of doing that, right? But if a person is not eating any carbohydrates, so they're not eating like things like pasta, rice, potatoes, these things which have carbohydrates in them, if you're not eating them, then your body will be low on glucose, and then your body will be thinking, okay, I need to have a backup plan. So what can I do? So the adipose tissue is that backup plan, because the body is always storing, fats into these adipose tissues, and then when you don't have the glucose available, the body is then going to try and take those fats and then basically break them down in order to still get energy, basically. So it's the body's backup plan. All right, so skin derivatives. So things which basically come from the skin, or things which can be found on the skin. So we've got scales, which are called squamae. They are dermal in fishes, meaning that the scales basically are found in the dermal area of the fishes. That was the second layer. Okay, so the scales of fishes are found in the dermal layer right here. And then the reptiles, their scales are on the surface in the epidermal area. Okay. You can find feathers on birds, which is attached to the skin, of course. You can find hair, which is coming out from the skin in mammals. And then the dermal derivatives, they're always covered by the epidermis or epidermal derivatives, because, of course, the dermal derivatives will always be found right here in this middle layer. And they include bony scales in fishes, bony plates in turtles, crocodiles, and certain extinct reptiles. Okay. So here are some groups of fishes and the type of scales that they have. So stenoid, cycloid, ganoid, and placoid. You're not, I don't think you're expected to know exactly, okay, what type of fish has got what type of scales. I mean, if you have the extra time to go over it and to memorize it, then of course go and do that. But it won't be a matter of, okay, pass or fail. You have to tell me. So Fatima, for example, you have to tell me the salmon fish, what type of scales does it have? Like, that's not normally a question that you would be asked. Okay. Now, the phylogeny of skin in early vertebrates. So the protovertebrates are the earliest vertebrates. And an example of them is the branchiostoma. Here you can see the branchiostoma. So the branchiostoma, remember how we said that the earlier vertebrates are kind of like bottom dwellers, filter feeding animals, or bottom feeders that just feed on like any type of things that they can find, kind of like scavengers. So these animals are very, very simple, and branchiostoma is one of them. And because it's simple, it only has a single layer of epidermal cells, as of course it's not a complex being. But then when you compare this type of animal to ourselves, later on you'll realize that we are like much more complex, and that is because we do many more things than a branchiostoma, for example, would be doing. This picture here, I would say, is important to memorize if you can, because this basically will help you remember the classification and kind of the groups that the animals are going to go into. So after you talk about the super kingdom, which is the bacteria, the archaea, and the eukaryotes, when you get to the eukaryotes, you have these basically. And of course, as we can see here, we're seeing peripheral right here, which was the sea sponges, and it's also the earliest primate, or not primate, but the most primitive. It's the most primitive animal or eukaryote within evolution. Okay. And then, of course, we have our vertebrates last. So it basically goes in this order. And then we spoke about protostomes and deuterostomes a little bit yesterday also. Does anybody remember what the difference was between them, between protostomes and deuterostomes? So it's got to do with the embryonic development. Yeah?
The restome, like it's the anus develops before the mouth, and
Yes.
then the protostome is the other way around.
Exactly. Very good. Very good. That is the exact explanation. Okay. Now, the evolutionary innovations in fishes when it comes to their skin. So, their epidermis is made out of living cells that have low keratin in it. Keratin is a protein which provides strength. And then the epidermis will basically cover their dermal scales. So remember we said that in fishes, their scales are actually found within their dermal layer, and then they have like a little bit of a epidermis covering on top of it. They have some mucus glands. Usually these are unicellular glands. The animals which first developed multicellular glands were the amphibians. So fishes have unicellular glands inside of their epidermis, and what they do is they basically, just like the hagfish that we mentioned yesterday, which was a type of agnatha, or a type of jawless fish, they can release these slimy kind of like a type of slimy substance, kind of like mucus, so that they become slippery, or their skin becomes slippery, so that they can avoid predators and escape, essentially. Some fishes have got venom glands, especially near their fin rays. And so you would see them in fishes that have this, basically. They've got like these rays here. Remember yesterday we spoke about the two groups of fishes, or bony fishes, which were the ray-finned fishes. So this here is an example of a ray-finned fish. And then the other example we had was the lobe-finned fishes that had like the skeleton, which kind of looks like the fingers, which looks like the bones of the fingers. So some of the ray-finned fishes, they have venom glands, where of course they're releasing venom again in order to protect themselves. Then the fishes have got pigment cells within their dermal layer. Again, a lot of the things are inside of their dermal layer. The epidermal layer, all it's got would be the living cells that have got the keratin in it to provide a little bit of strength. Then the sensory organs. So we've got neuromasts and electroreceptors, which are found in the skin of the head and along the lateral line. So when we're saying lateral line, what we're talking about is like, imagine you split the fish in half, but like from side, like basically from this side, like you're halving it in this way. So you have like a lateral line basically there. I'm not sure if I have a picture of that in this slide, but I think it's coming up. That's fine. Okay, so the evolution of scales. Now, okay, I need to send this presentation, not presentation, but I need to send this page to, okay, cool. But sorry, so the evolution of scales. So where did scales come from? And this is actually very, very interesting, because it's believed that our teeth, so the structure of our teeth is identical to the structure of the scales which cartilaginous fishes have. So cartilaginous fishes, like sharks, for example, they have placoid scales. And if we just look at the structure of the tooth, like the inside structure of it, we normally have a enamel on the surface, and that enamel is like that hard protective barrier. And then inside we have dentin, and dentin is a little bit softer. And then we have the pulp, and the pulp basically is where you have the nerves or innervations, basically. So the nerves and the blood supply. Okay, and this basically supplies the dentin so that your dentin is nice and healthy. And the placoid scales that the sharks have literally have this same exact structure. So they also have a enamel on their placoid scale, and then a dentin, and then the pulp. Okay. So the early vertebrates, they had massive bony exoskeleton, but then as they became modern fishes, first of all, they lost it as cyclostomes. Cyclostomes are agnata. So agnata basically lost the bony exoskeleton, and as we know, agnata, or the jawless fish, are cartilaginous. Okay, so they also developed reduced dermal scales, or not the agnata actually, the modern fishes. Now the modern fishes have got less. So before the older fishes, they had a lot more, and then now they have a lot less. And we spoke about this already. So we basically said that the placoid scales, which can be found in cartilaginous fishes like sharks, their placoid scales are homologous, meaning that they're basically the same in structure when it comes to the teeth of the vertebrate. Okay, now the evolutionary innovations in amphibians. So amphibians have, first of all, a cornified epidermal layer. So this just means that their epidermis layer is basically a little bit harder, a little bit tougher. And the purpose of this is to protect the body from dehydration. So even though they are amphibians, they are still trying to make sure that they are not, of course, becoming dehydrated. I mean, their skin is always moist, and this is to help them with the respiration. But they are, just trying to be on the safe side also. So that's why they have a cornified epidermal layer. Now, they also have multicellular glands, as we mentioned. Some of these glands can release poisons in order for them to, or venom in order for them to protect themselves. And then some of these glands basically produce things like water onto the surface so that they can keep their body surface moist, and to basically allow that gas exchange to happen through their skin. And these multicellular glands can be found in their dermal layer. Okay, and so we mentioned that they've got cutaneous respiration. So cutaneous basically just means like the skin. Okay, so this is important for most amphibians. They have well-developed set of capillaries in the dermal layer, and of course these capillaries are going to help with gas exchange. Okay, they also have the capillaries within the deep layers of the epidermis. Okay, so essentially, if we just go back to the picture, so we're saying basically that they have a lot of capillaries, or a very, what's it called? Yes, they have a lot of capillaries basically within their dermal layer, and also some within the bottom of the epidermal layer. Okay. As we said, their glands can also secrete poisons so that they can protect themselves from predators, but not all amphibians have this, only some of them do. So here we can actually see the multicellular glands. Now, chromatophore, you might be wondering what that is, but that's basically a pigment. Okay, chroma basically means color. So that is pigment, which basically gives them the color that these different animals, or these different amphibians, would have. As we said, they have their mucus gland right here that would secrete the substances which would make their skin moist. And then they have their poison gland to deter away from the predators. Okay. Now the evolutionary innovations when it comes to the amniotes. So the amniotes we mentioned are like reptiles and birds. Now the reptiles, they have a stratum corneum. So this is within their epidermis, okay? So within their epidermis, they have a stratum corneum, which is the top layer. The stratum corneum is best developed when it comes to the reptiles, because it is adapted in a way which allows them to protect themselves to like a full level. They can protect themselves from water loss. Okay, and this is very, very important, because we mentioned before that the amniotes and the reptiles were the first group to kind of venture away from bodies of land. Okay, so they had to figure out ways, or their bodies had to adapt basically to ways in which they would have to decrease any water loss, because they'd be away from bodies of water for a long time. And one of those adaptations were the development of the stratum corneum. So they have a dry epidermis. they don't have many skin glands, and if they do have skin glands, they would have the scent glands around their mouth and their cloaca. The cloaca is basically around their anal region, and this is so that they can recognize sexual partners and then they can mate and partake in reproduction. Okay, now squamate reptiles, these would be lizards and snakes, and crocodiles and turtles have different structures when it comes to their skin. So the lizards and snakes, they have thin overlapping horny scales, meaning that the scales are very they can be kind of prickly, they protrude outwards. And then the crocodiles and the turtles have something called scutes, horny scutes and plates. So where the lizards and snakes have scales, the crocodiles and the turtles would have plates instead. Okay. And they cover the bony plates that come from the dermis layer that are underneath them. Okay. So then that means that these plates are actually found in the epidermis, in the epidermal region. Okay, now the mammals. So in mammals, the stratum corneum, which we said was the top layer, it becomes a little bit thinner, okay? And we develop hair for the first time, as we said, and then this hair is known as a horny derivative, as it's basically supposed to be, what's it called? Because of the structure and the way that it protrudes out of the body. And it is homologous to horny scales in reptiles, meaning that the structure of the hairs are similar to the structure of the scales in reptiles. Okay, and here you can see the hair kind of emerging out or erupting out, specifically right here from the last part of the dermal layer. Okay, and it protrudes outwards. It goes through the dermal layer and out into the epidermis. Okay. So the epidermal composition. So this is when we're talking about everything to do with the epidermis. So the epidermis in mammals is stratified. So remember we said that the branchiostoma, which was the early protovertebrate, it only had a single layer of cells within its epidermis. Well, mammals have got stratified, they've got basically stratified epidermis, meaning that they have lots of layers. And these layers are basically, they are the stratum corneum, which is the outermost layer, the stratum lucidium, stratum granulosum, and stratum spinosum, and then stratum basale. And each of these have got different roles. So starting with the stratum corneum, you have a dead layer of cells right here at the top. Okay, and these cells are specifically keratinocytes, which would basically be cells which would release keratin. Then you have the stratum lucidium right here. You have the stratum lucidium after the corneum, and this is a transparent layer, again composed of dead cells. Then you have the stratum granulosum that has cells with granulated cytoplasm, which will be starting to lose their nuclei. Then when they lose their nuclei, that is, they basically are going to go to the top. So I know it's numbered 1 to 5 in this way, but it's supposed to be the other way round. So it's supposed to be stratum corneum first and then the stratum basale after. But in the granulosome, when they start to lose their nuclei, is when they are going to start to basically become dead or thought of as being dead. The stratum spinosum, so the next structure, have got cells that look like they have spines because of how they kind of are interacting with one another. And then you have the deepest layer, so the stratum basale, and this layer is actually the layer that has the activated and alive cells, which are constantly dividing. And what they're doing is the stratum basale is responsible for constantly renewing the layers above it. Okay, so it's renewing the layers above it. And this is important because, as we said, the skin has got a mechanical function, so it has to be highly regulated, okay, to make sure that these dead cells are sort of like, they're sent up to the top, and then, you get rid of them. Okay. Now, in the epidermis, you can also find melanocytes, and these are basically cells which release and produce melanin. And you can also find Langerhans cells, which are helping with our immune system, and they're basically helping to defend against any pathogens that shouldn't be there. And yeah, actually that's fine. All right. Then the hairs. So hairs are found in most mammals, and they could be modified into being bristles or spines in some species. As we know, they provide insulation. They can also help with camouflage, and they can also help with skin mechanoreception. Mechanoreception. Does anybody know what mechanoreception is? Or any guesses as to what mechanoreception would be? If we said that it's something that you can, that the hairs are helping with, yeah, in the skin.
Is it receptors for stimuli and others
Yes, it is. But what type of stimuli?
external?
Yes, but like, could you describe it? What kind of stimuli would they detect?
Maybe changes in temperature or touch, physical touch.
Well, touch. Yes, touch is more correct. So basically, they'd be able to detect like pressure. Okay, so the hairs that we're saying that are on the skin are able to detect when there would be some pressure in some mammals. So some mammals, like cats, for example, have got whiskers around the eyes and the mouth, and they serve as tactile function. And basically, I don't know if you know, but cats basically, they use their whiskers in order to figure out if they can go through tight spaces or not. Because they basically realize that if their whiskers can fit through an area, okay, then they assume basically that the rest of them, like the rest of their body, can also go through that area. Okay. These whiskers possibly evolved first for this purpose in mammalian ancestors, for them basically to have that tactile function and to figure that out, to figure out the proximity of things. Now, some mammals also have got claws or nails or hoofs, and this is basically to help them with their either to help them with their locomotion or to help them defend against predators. Function of hairs now. So we said insulation and camouflage, mechanoreception, and tactile function. Okay. Now the dermal layer. So the dermal layer isn't as complex as the epidermal layer, where in the epidermal layer we had 5 parts or 5 layers. The dermal structure has got only 2 layers. So we have the papillary layer, and then we have the reticular layer. So the papillary layer is the first one, and it's the one that's right underneath the basal layer of the epidermis. And then the reticular layer is right underneath that one. It's right underneath the papillary layer. So what is the dermal structure composed of? So basically it has the connective tissue. Okay, so this connective tissue is produced by fibroblasts, and it's basically there to support the epidermis, to support the endothelial cells in the epidermis. There are sensory corpuscles within the dermal layer also, and this is important for sensation. There's blood vessels, of course, the smooth muscle fibers, hair follicles also grow from there, and skin glands. The skin glands could be sebaceous glands or sweat glands. The sebaceous glands, they partake in holocrine secretion, and that basically means that when the oils, so the sebaceous glands would release things like oils and stuff, when they're releasing this, the whole cell is going to disintegrate. The whole cell is going to basically break down in holocrine secretion. And then the sweat glands that are releasing sweat to cool us down whenever we're feeling too hot are either released through merocrine, in a merocrine way, or through apocrine secretion. And when it's merocrine, nothing actually happens to the structure of the cell. The cell remains intact, and it's just able to release the substance easily, like without the cell breaking down. But then the apocrine ones, they break down parts of their cytoplasm. They break down parts of their cytoplasm as they're releasing the secretion up into, like, out from the dermal structure and up into the epithelial layer within the epidermis. And that is something that was covered in cytology before. Okay, so the mammary glands are unique to mammals. The mammary glands are called glandulae mammae. They are modified sweat glands. They are always paired. You don't need to know how many of them there are usually, but you could just say that they are paired. The number of them and where they may be will be different in every species. Now, just as an example, there's some information here. So primates, and elephants, they have single pairs of mammary glands where the thorax is. And then most herbivores, so plant-eating animals, they have one or two pairs of mammary glands. And then mammals with large litters, basically meaning mammals that are going to give birth to many live young at a time. This would be rodents, carnivores, and pigs. They will have, of course, a lot more mammary glands so that they're able to provide for their, provide milk for their young. So, yes. So basically, the amount of mammary glands depends on how many live births they can give in like one sitting. Okay. Now, the development of the melanocytes. So we said that the melanocytes are the cells which are going to release melanin. They themselves come from the ectoderm, so the outer germ layer. And then they come from the neural crest, and they go from the neural crest to the epidermis. And in mammals, you can find the melanocytes within the epidermis, but then the pigments within the fish and the amphibians, you find them in the dermal layer instead. So active melanocytes are initially present throughout the dermis in embryonic development in mammals, but then they're going to go up. They're going to go from the dermal layer to the epidermis. And some of them can go through apoptosis, so cell death by the time of birth. And then, of course, the amount of melanocytes we have determines the amount of melanin that we will have. And of course, the more melanocytes somebody has, the more brown they would be, or the darker they would be. Now, the developmental stages of the epidermis. So how does the epidermis actually develop? And then we've got the hair follicles, and then we're done with the syllabus point. So the formation of surface ectoderm. So the ectoderm initially forms a single layered epithelium. Okay, so single layered, similar to the first vertebrate that we spoke of, that only has a single layer of cells in the epithelium. So that is how the epidermis first formed. Came from the ectoderm and was a single layered epithelium. Then something called a periderm formed, and the purpose of this periderm is basically to provide a protective layer on top of the epithelium. Okay, and it's there until the epidermis fully develops, and until the epidermis basically becomes cornified, meaning it becomes stronger. Okay. Then there is epidermal stratification, meaning that the epidermis starts to become stratified, meaning that it becomes layered. So it goes from being one single layer to multiple layers. How many layers of epidermis have we got? Does anybody remember? Okay, we have 5 layers. 5 layers of our epidermis. The most bottom one being the stratum basale, and then the top one being the stratum corneum. Does anybody remember how many layers we have of the dermis?
Yeah, we have 2 main layers.
Yes, very good. We have 2 main layers. Okay, and I will tell you guys, because I don't expect you guys to have remembered the name. I just wanted to know the numbers. So the 2 dermal layers are the papillary layer, and then we have the reticular layer underneath it. So the papillary layer and then the reticular layer. Okay. Yes. So then cornification. So for the cell or for the skin to become tougher, or for the skin to become cornified, we need to have dead keratinocytes at the top. And these dead keratinocytes are basically, as we said, cells which produce keratin. And they're held together by other proteins and lipids that will keep them there. And as we said, they're there to protect the skin. So either mechanically or biologically by protecting the skin from pathogens. So the permeability changes. So during development, the skin is of course going to change. So fetal skin is highly permeable at first, and this is so that the embryo, the extra embryonic membrane, which was the amnion, can basically determine how much amniotic fluid it needs to produce. The amniotic fluid is then going to surround the fetus, right? That amniotic fluid, what was the purpose of it that we mentioned yesterday? What does the amniotic fluid do? So we mentioned the amniotic fluid. No, the amniotes are named after the amniotic fluid, right? Which you can find that surrounds the fetus during embryonic development. So what is the purpose? Why do we have that liquid there? What do you guys think that liquid is doing?
So it mainly protects the fetus itself,
Yep.
and it helps with other things. It helps with the waste exchange between the
Ah,
fetus and the mother. It
okay.
helps with
Oh,
barriers, with temperature regulation.
okay. Okay. So the amniotic fluid, yes, it basically is able to absorb, like shock movements. So basically, it's kind of like a cushion, like it cushions the baby and protects the baby from, the movements of the mother or anything like that. So that because at those stages, as the fetus is developing, of course, it's like very vulnerable, and it needs that watery layer to absorb any shock movements, and to cushion it, to help with any potential damage. So the amount of amniotic fluid that is released by the amnion, which is the extra embryonic layer, or the not layer but the wall, that is determined by so basically the permeability of the fetal skin basically helps to determine how much of that amniotic fluid should be released. After keratinization, which basically means the cells become keratinized, the skin then becomes impermeable, because after that you don't need the amniotic fluid anymore. So we said that the periderm is a protective layer that goes over the epidermis as the epidermis is developing. But towards the end of the epidermal development, the periderm is going to detach itself because it's then done its function. Okay. Then we have something called the vernix caseosa. Now, the vernix caseosa comes from the periderm, so the detached periderm. It's kind of part of the periderm, and the whole point of it is to basically provide a greasy coat around the skin to sort of protect the fetus from the amniotic fluid, because this amniotic fluid is, of course, good for the fetus in the sense that it's protecting the fetus from from any sudden movements. But at the same time, this amniotic fluid could go through the skin of the fetus. And as we said, the skin of the fetus is highly permeable, and you don't want like a large amount of amniotic fluid to go through. Some amounts is okay, but not like a large amount. So that's what the vernix caseosa does. It stops that. Okay, then the development of the hair follicles. So they first form hair placodes. So the epidermal keratinocytes, they form clusters. These clusters then become larger, and they, become longer, and then they generate what we know as hair placodes. Then after that, there are specialized fibroblast clusters that are above the placode. These fibroblasts, they basically help. Remember, we said that the fibroblasts are helping in developing the dermal layer. and basically they're developing their connective tissue within the dermal layer. So the keratinocytes that we formed and the fibroblasts are going to work together, and they're going to increase in number. They're going to proliferate. Then we're going to form the hair germ. And basically with this, what's happening is that there's going to be downward growth of the epidermal part, and this will shape what we know as the dermal papillae. So after this, we form something called the hair germ, and then the keratinocytes, which are of course producing keratin, are going to penetrate through the dermal layer, okay, and make the dermal papillae. So all of this basically is starting off first in the epidermal layer, okay, so the bottom part of the epidermal layer, and then it's going to protrude into the dermal layer. So it's starting in the epidermis, and then it's going to go down into the dermal layer. The placode that we mentioned is right here. Here you can see the placode. So the cells are going to multiply, multiply, multiply. They're going to make a primary germ center, as you can see here, as they're going downwards, okay? And they continue to go downwards until they form this matrix of cells right here, okay? And then here later, they're going to have some capillary loops, and then essentially, eventually what's going to happen is we're going to form the outer root sheet and the inner root sheet. So right here, which is then going to eventually give rise to the hair. Okay, so this hair is, by the way, also made out of keratinocytes. So our hair is made out of keratin, essentially. Okay. Now, any questions before we move on? I think it might have been a lot of information, but is anybody like completely lost? No? Okay, so basically, just to summarize, just very quickly then, when we're talking about the skin, you first have to talk about the functions of the skin, what the purpose of the skin is. Then you're going to talk about like appendages that can come with the skin. So things that come out of the skin, like we said, hair and fur. And then, of course, birds have feathers. And then you'll have to also talk about the differences in the skin of amphibians and the reptiles. We said that the reptiles have the best stratum corneum. We said that the stratum corneum is the outermost layer of the epidermis. We said that the epidermis consists of 5 layers. Okay, I would suggest that you learn those 5 layers. And then we said that the dermal layer consists of 2 layers. Please also learn the names of those 2 layers. We said that the fish have got scales for protection, and these scales are basically found within their dermal layer rather than their epidermis. We said that their epidermis consists of some living keratinocytes, which is different to mammals because our skin, like our outermost part, so our stratum corneum, is made out of dead keratinocytes, whereas the fishes, their top layer is made out of living keratinocytes. So there's a difference there. What else was there to mention? So we said that the amphibians, they have to keep their skin moist. The multicellular glands appeared for the first time in amphibians, okay, to keep the skin moist. Why does the skin of the amphibians need to be moist? Guys, why does the skin of amphibians have to be moist? What's the purpose? What does the amphibian skin do? What can they do through their skin?
So they can, they basically, they need it for oxygen. They breathe and they respire through the skin.
Exactly. So they respire through their skin. Amphibians respire through their skin. Guys, this is very important to remember, okay? They respire through their skin. Gas exchange basically happens through their skin. And for this reason, the skin needs to be moist. If their skin was dry, then this gas exchange would basically be impossible. Then the reptiles, they don't need this function because reptiles have better use of their lungs. They use their lungs better. because they have the reptiles, they've got the intercostal muscles, which happens for the first time in reptiles. And these intercostal muscles, they basically allow for the expansion of the lungs, right? So that all the gas exchange that the reptiles need to do will only happen in the lungs of those reptiles. Whereas the amphibians, they need that extra help because they don't have the intercostal muscles. So their lungs can't really expand, basically. We said that the reptiles have got very dry skin. What was the purpose of reptiles having dry skin? And Adnan is not allowed to answer. What is the purpose of reptiles having dry skin, guys?
Does it reduce water loss so they can live in areas which don't have water sources?
Exactly. And the whole purpose of that is because we said that the what we're trying to do is we're trying to reduce the dependency on water, because these animals are going from living a aquatic lifestyle to living a terrestrial lifestyle. So they have to adapt to harsher environments once they're on land, because not everywhere in land has got water. Okay, so these reptiles, they have to do their best to reduce water loss, as they don't know when they're going to be coming across the next body of water, essentially. Okay, so those were the things to mention about the development and the phylogeny of the skin. Guys, I need you to answer more of the questions. Otherwise, I'm going to start to get really annoying and I'm going to start to pick people. So do pay attention and don't be nervous, okay? Just answer to the best of your ability. And if you get things right, then that shows you, okay, cool, you've understood it. If you get things wrong, at least I can give you the correct answer, and then it's a win-win situation regardless. If you get the answer wrong, if you get the answer right, you are still benefiting. Okay, so please do answer the questions. So now the phylogeny and the development of the nervous system. So first, we have to talk about the overview of the nervous system. So very simply, the nervous system is made out of excitable nerve cells called neurons, and then we have the synapses, which are the gaps in between these neurons, right? Then we have glial cells, which support functions like myelination and ionic regulation. Does anybody know what myelination is? I mean, you should know, actually. So what is myelination? This word right here, what does this mean? Or where do you find myelin?
Formation of myelin sheet around the nerve fiber.
Exactly. And what is the purpose of it? You know?
To improve conduction.
Exactly. Very good. Okay, so the neurons and their functionality. So they vary in structure, but they operate based on excitation or inhibition. Okay. Now, let's we skip over actually. So the subdivisions. So we have the central nervous system and then we have the peripheral nervous system. So the central nervous system includes the brain and the spinal cord and is the body's control center. And it has centers for sensory, motor, and data integration. Then the peripheral nervous system is composed of the spinal and the cranial nerves, and it links to the central nervous system. And then it has sensory receptors that process environmental changes. So now the functions. So we have sensory input, and this is basically when nerves are getting impulses from sensory receptors, and they will then send them to the brain and the spinal cord. Then there's integration, where the brain will process this information. Then there's the motor output, where the impulses are sent from the brain to the spinal cord and then to the muscles and the glands, so that the muscles and the glands can perform an action. And yeah.
Yeah, sorry to interrupt. I actually can't see your screen. Not sure
Okay.
why, but
Is it just you or is it everyone? Can you
Yeah,
all see?
me too.
You too? Okay. All right, I will stop sharing and share again. There we go. Okay, can you guys see right now or no?
Yes.
Yes, that's better.
Okay, cool. All right. Then there's this interaction. So the nerves from the PNS will connect with the CNS and vice versa, so that there is coordinated function all around the body. Okay, so we did the overview now of the nervous system. Now we have to talk about the phylogeny of the nervous system. So cnidarians have a neural network which is very diffuse. So this is actually one of the stop test questions where it was, I don't remember how exactly it was phrased, but it was something about which animal group has got a diffuse nervous system. So the animal group that has a diffuse nervous system would be the cnidaria. Now, the cnidaria are animals like the jellyfish, okay? Then we have annelids, which are worms. They have segmentation, and then they have a ventral central nervous system, and they have ganglia to help with sensory cell localization. Their cerebral ganglia helps them with some level of vision and olfaction being smell. The flatworms have got sensory cells which are concentrated in specific regions. Then their nervous system has something kind of close to a central organization. And yes, and that is all that is important to note for now. This image would be important for you guys to remember, okay? Because this image basically is talking about how the brain develops and what structures come from what, essentially, okay? So just ignore the two images on the sides, so the lateral views, ignore them, but you should learn this image, okay? So I would suggest that you draw it out, you know, like if you've got like a whiteboard, draw it out and then, you know, rub it out and then draw it and then erase it and so on, so that you can remember what comes from what, okay? Now, the evolution of the brain. So cyclostomes, which are the agnata or the jawless fish, for example, lamprey, they have distinct parts within their brain. So their telencephalon, which was this structure right here, which comes from the forebrain. Okay, so the telencephalon, which comes from the forebrain, is going to develop or become the olfactory brain, meaning that it's going to be the area which is going to help with some level of sense of smell. Okay, and then they have their mesencephalic tectum, which is another area which is going to help them process visual information. Then their cerebellum is a bit underdeveloped. Now, the cerebellum is the area which is important for movement. Now, the cerebellum of the agnatas is underdeveloped, and they also don't have paired fins, meaning that the agnatas are not very good swimmers, okay? They don't really swim well. They kind of maybe, the word wouldn't be glide, but they don't really swim well, okay? They just have their caudal tail, which is that one tail at the end that will just push them through the water, but they're not really good swimmers, okay? So it makes sense that their cerebellum is underdeveloped because the cerebellum is important for movement. Then cartilaginous fishes, remember, they're much better swimmers, and for that reason, they're going to have a well-developed cerebellum. The cartilaginous fishes also now have paired fins. Their tectum and then their telencephalon are better developed, meaning that their sense of smell is better. The olfactory bulbs are larger, and they're basically larger. Then the bony fish, they have optic lobes of tectum, which becomes enlarged, meaning that their sense of vision is going to be better, okay? And their sense of vision is going to overtake the level of olfaction. So the sense of vision is basically going to be better than their sense of smell. Okay. Then the evolution of the brain when it comes to the amphibians. So the cerebellum is now going to become smaller and simpler because of their lifestyle. As we said, cerebellum is important for movement. The amphibians, of course, are still moving, but they're not swimming, okay? So for that reason, it becomes a little bit simpler. Their visual perception becomes much better. In fact, their eyes move. So the fishes basically have eyes laterally, so on one side, right? So they have their eyes on one side and then the other eye on the other side. But then the amphibians, they have their eyes on top of their head, and this also, of course, helps their vision. Then the amniotes have elongated and better developed cerebellum because they're going to do more movements. Their optic lobes are enlarged compared to amphibians, meaning that their sense of vision is much better. And then their telencephalon becomes the largest division. And then here we have a picture basically comparing a frog brain and a dog brain. Of course, the brain of the dog is going to be bigger as the dog would be doing more complex activities rather than the frog. Okay, then the mammalian brain. So our brain is, of course, the most complex. We have our cerebrum, which is the most important part of the brain. And then we have both our hemispheres, which are connected by the corpus callosum. The olfactory bulbs then are well differentiated in most mammals, meaning that most mammals have a much better sense of smell, and we are able to breathe through our nose much better compared to the earlier animal groups. In some groups, the olfactory bulbs are smaller and covered by the cerebral hemispheres. So, for example, in the primates. Okay, then the primates they have their olfactory, basically, their olfactory system is diminished compared to their optic lobes. Basically, they have, again, better vision than smell. And then the cerebral cortex is smooth in many mammals, but you form gyrae in large and advanced mammals. The gyrae is basically the grooves which you can find on the brain. The gyrae increases the surface area and the amount of gray matter that we've got within the spinal cord. The area called the neocortex will become highly developed and is subdivided into areas, especially in primates, because if you remember, primates have got better level of, not even better level, but they have more social interactions or more complex behaviors. And this is why the neocortex will be more developed. Now, because of this, because of our larger brains, we have some negatives, okay? For example, we have high metabolic expenses, meaning that because our brain is larger, we require more nutrients in order to power it, basically. Okay, and we'll also have slower reproduction than animals that have smaller brains, because again, reproduction requires a lot of energy, and our brains are larger, so we need to give it more energy, essentially, for any simple function to even occur. Other mammals will have simpler brains, and when they have simpler brains, they could have shorter parental care, so they're looking after their offspring for a shorter amount of time, and then they would have faster reproduction, so that they wouldn't have to trade off their slow reproduction and their high metabolic expenses when they have bigger brains, basically, or more complex brains. Okay, now the structure of the spinal cord. So the structure of the spinal cord it basically looks like this. Okay, so we have dorsal horns at the top, then we have the ventral horns here at the bottom. So the dorsal horns, they form dorsally, meaning they form from the back, and they consist of left and right grey matter, and they receive sensory dorsal roots of spinal nerves from the sensory ganglia. Then the ventral roots, the ventral horns, which are at the bottom, They are also made out of the left and right grey matter, and they send out motor ventral roots of spinal nerves. So this grey matter and white matter that we're talking about, the grey matter is in the middle, so it's this X shape. Then the white matter is this pink area. And they have a difference in color based on what's in them, okay? And they're also different based on their functions. So the gray matter is made mainly out of cell bodies of the neurons and dendrites and unmyelinated axons, whereas the white matter is made out of myelinated axons. So these myelinated axons are basically fatty substance, made out of fatty substance called myelin, and this fatty substance is white in color. And because it's white in color, that's why the white matter appears to be white, and that is why it's called white. Whereas the gray matter, in comparison or in contrast, doesn't have this myelination. So because of that, it's not exactly white. Instead, it has the cell bodies, as we mentioned. So basically, it's got the cell bodies and the axons and not the axons, sorry, but they've got the cell bodies and the dendrites. And that's basically what makes it look grey. So the grey matter is important for processing and integrating information that you get from the sensory organs and to also initiate motor commands. Whereas the white matter is basically, it transmits nerve impulses between different regions of the brain and the spinal cord. Now, the specializations in different vertebrate groups. So when we're talking about the spinal cord of different vertebrate groups, what do they look like? So the jawless fish, they have a flattened ribbon-like spinal cord. They have an absence of horns. So you see how we can see the horns here? They don't have this. They don't have these horns. And because of that, they're very, very simple beings. Then the fishes have got dorsal horns, which are very close together. So this is basically what a fish's spinal cord would look like. They have these dorsal horns, which are very close together, and their spinal cord goes through the entire length of the vertebral canal. So, of course, as we know, the vertebral column or the vertebral canal contains the spinal cord and protects it, right? So in fishes, we're basically saying that the spinal cord goes all the way through the vertebral column. Now, this is different to mammals, which we're going to talk about now. So mammals, they basically have a spinal cord which is shorter than the vertebral column. And this basically happens because of early kind of stopping of neurogenesis. So neurogenesis, which is the formation of the nerves, will stop at an early point. And when it stops, well, the spinal cord will also kind of abruptly stop too. And that's why it's not really matching completely with the vertebral column. The tetrapods, so the amphibians, the reptiles, the birds, and the mammals, they have broadening of the spinal cord at the neck and at the lumbar area. And the reason for this is to increase control over limb movement. So again, we said that we have kind of protrusions or larger areas of the spinal cord at the neck and at the lumbar area. Okay, so basically cervical area and the lumbar area. Now, the roots of the last spinal nerves are going to go into an area called the vertebral canal, and then it's going to form something called the cauda equina. Okay, and this cauda equina is going to basically do its best to try to reach some of the body segments. So in summary, what are we saying? So we're saying the earliest vertebrates, so the agnatas, the jawless fish, their brain is the most simple. The brain only serves basic functions which they need for survival. As the vertebrate groups diversify and we get more of them, the brain structures, of course, are going to be more defined and more specialized, because we're going to go from animals which are very simple to animals which are very complex. And this is to increase their survival and to increase their lifespan. The cartilaginous fishes have a more developed brain with distinct areas for sensory and motor processing, but it's still very simple compared to other advanced vertebrates. When the bony fishes came, the brain differentiated more to accommodate for the aquatic life and the swimming behaviors. Then the amphibians transitioned to land, and they went through significant brain development again to help them adapt to the terrestrial environment. And specifically, what happened with the amphibians is that their sensory processing abilities became even better. I wanted to say an example with the sensory processing ability of the amphibians would be that the ears of the amphibians became better developed as they developed a middle ear and they developed a eardrum, whereas the fishes that were before the amphibians, they only had the inner ear, meaning that their sense of hearing wasn't really that developed, wasn't really that advanced, because they didn't really need much of a sense of hearing, because they basically are able to feel what's going on around them through their skin. Now, yes, so the reptiles have even more specialization and enlargement of the brain regions, and this helps them to adapt to their habitats and the different behaviors, such as hunting and building a nest. As the birds became the first animals to be able to fly, their brains became, of course, more enlarged and more complex, specifically in areas that are associated with vision and motor control, so that they can, of course, spot their prey. And the motor control, of course, is important for them to be able to control their wings so that they're able to fly, basically. Mammals then have the most advanced brain structures with highly developed and differentiated brains, and this allows us to live in different habitats and have complex social behaviors. And this can be seen through the expansion of our cerebral cortex. So our cerebral cortex is the most developed within mammals, especially in primates. And we also have advanced sensory perception and, of course, cognitive abilities. So meaning that we're able to form thoughts. And this all comes with the advancement, or the not the advancement, but the change of the cerebral cortex, the development of the cerebral cortex. Does anybody have any questions in regards to this topic? Or any questions at all, really? No. Okay. All right. Now we're going to talk about the sensory organs. Okay. I mean, we were talking about some of the sensory organs, but we're going to be talking about the skin sense organs. Okay. So the sensory system is an important part of the nervous system, as it is basically able to receive external and internal stimuli and then respond accordingly. These systems include sensory receptors that will detect stimuli, which are the neural pathways that will transmit information, and then also the specific brain areas that will analyze it. Receptors can be either simple nerve endings or complex sensory cells, and they come from the ectodermal layer of embryonic development. So the ectodermal layer being the outermost layer of the germ layers. These receptors can be classified based on their location, the type of stimuli that they detect, and the type of stimuli that they will respond to. So skin reception, or skin sensation, I should say. Skin sensation involves general receptors which are either unencapsulated or encapsulated with nerve endings that are underneath the skin surface. These receptors will connect to neurons that can be found in the sensory ganglia. Then we have the types of cutaneous receptors. So these are the types of receptors which are found in the skin. So there's 4 main types: the mechanoreceptors, thermoreceptors, nociceptors, and the chemoreceptors. All 4 of them are adapted to basically detect different types of stimuli. So the mechanoreceptors will detect mechanical pressures. So examples would be the Merkel cells, which detect fine touch, and then the Pacinian corpuscles, which detect vibration. Then we have thermoreceptors, which are sensitive to changes within the temperature. We have noise receptors, which basically detect pain, so that we can detect tissue damage or potential harm. Then we have chemoreceptors, which are more associated with taste and smell, but some skin receptors could also react to chemicals, such as the chemicals that are released by damaged cells. The receptors respond to external stimuli by either directly making a nerve impulse or indirectly by synaptic transmission if they are not neuronal cells, basically. If they're non-neuronal, basically meaning if they're not on the nerve. So if they're on the nerve, then they will directly generate the nerve impulse. If they're not in the nerve, then they're going to do it through synaptic transmission. Now the neuromasts. Neuromasts can be found within aquatic vertebrates, and they are specialized mechanoreceptors. So they can be found within fish and some amphibians. They're important for detecting water pressure and low frequency sounds, and this will help them in.
I'm so sorry. I can't see your slide that you put on the screen, and please ignore the noise in the background. I've got some maintenance people right now at my place.
No, it's okay. I don't understand why it keeps disappearing. That's so strange. No, thank you for telling me. And 1 sec. Can you see it now?
Yes, I can see it now. Thank you so much.
No worries. What was I saying? Yes, the neuromasts. So the neuromasts are mechanoreceptors, which you can find in aquatic animals, so basically fishes and some amphibians, and they are mechanoreceptors, meaning that they are detecting pressure, so water pressure, and to some extent, low frequency sounds. And this will help them in navigating and basically figuring out where they need to go, and spatial orientation, which basically lets them know where they are, essentially. Now the structure and the location. So the sensory receptor cells in neuromasts have got kinocilium and multiple stereocilia. Now these are structures which kind of seem like hair, okay? And these structures, they have some mobility, okay? In some cases they have mobility, in some cases they may not have mobility, but basically the neuromasts having these structures, so having the kinocilium and the stereocilia, basically means that whenever something pushes down on them, okay, that basically is what's going to start the nervous impulse. Then there are support cells, which are around the hair cells, and they provide structural support, and they keep the kinocilium and the multiple stereocilia there. And then the neuromasts can be found superficially under the skin or inside of canals so that they can be protected. Now we have the lateral line system. So the canal neuromasts are arranged along the head and the body within the lateral line system. And the lateral line system basically allows the fish to detect water currents and vibrations, and they need this in order to realize, okay, whether there is a predator near them or not. Okay, so the lateral line system, I think I had a picture. It's the trunk canal system, but the lateral line system is technically there. So here, you can see the lateral line here. Okay, this is what's known as the lateral line right here. Okay, that's the lateral line. You can see that in fish. You can also see it in the amphibians right here. So this lateral line, basically what we said is, consists of neuromasts, and these neuromasts basically are mechanoreceptors, which will detect changes in water pressure. So they're able to detect whether there is a predator near them or not. Okay, so how neuromasts work. So this is a screenshot from the lecture slide. The neuromasts can be superficial or they can be found in canals, and these canals basically are invaginations inside of the epidermis. So invaginations just means like pits inside of the epidermis, and they're there sometimes so that they can basically have better protection. Now what essentially happens is that there's going to be some water displacement, and this water displacement is going to bend this structure here called the capula. And this capula, inside of it, has got the kinocilium and it's got the stereocilia. And underneath it, we can see, right here, this is where the stereocilia and the kinocilium will be. And then there is support cells around the stereocilia and the kinocilium. So the water displacement is going to bend the capula, okay? And then it's going to basically cause like a slight bending of the hair cells also, and then this is going to generate the stimulus, okay? Now, the consequences of a damaged lateral line. So when the lateral line system of the fish is compromised, it will of course affect their sensory capabilities, and it will then of course affect their survival. So fish have got like a schooling behavior. So I don't know if you guys have watched Finding Nemo, but there's basically like, groups of fishes that are always together. And when they're always together, you basically call this a school of fish. So basically, fishes, they rely on this lateral line to detect movements and positions of other fish which are in their school. Okay? Damage to this will basically cause issues with their coordination, and they won't be able to move as a group. Okay. The other thing is that the lateral line, of course, as we said, will also let them know if there is a predator close to them. Now, if this lateral line is damaged, then they won't be able to feel that difference in water pressure, and they won't be able to feel any vibrations, meaning that their likelihood of survival basically decreases. The lateral line also helps them in finding food sources, as again, if they are not prey themselves and they are predators, then again, they can use this lateral line to basically feel this displacement of water around them. And in this way, they are again able to feel where their preys are. Now, if they have this damage to the lateral line, again, they will not be able to find this, they won't be able to find their prey, basically. And of course, this will again affect their survival. So the role of electroreceptors within fish. So some fish species enhance their environmental perception through electroreceptors, which are another type of sensory receptors which are found in the lateral line system. So we said so far the lateral line system has mechanoreceptors, which are called neuromasts. The lateral line system also has the electroreceptors. These electroreceptors are able to detect electrical fields in the water, which are given out by many living organisms because of their muscle activity and their nerve impulses. Okay, so they can actually feel this. They can feel this coming from other fish, and because of this, they will then be able to find their prey easier because of these electroreceptors. So in areas where it's very like dark, or the visibility is very, very low. I mean, their vision is not the best to begin with anyways, but in areas where their vision is impaired, they can use this lateral line system that has the electroreceptors in order to find their prey very easily, even if their prey is well camouflaged. Now, while neuromasts primarily detect mechanical changes like water flow and pressure, and the electroreceptors, they give crucial electrical data. Together, these systems will basically give the fish a comprehensive sensory map of their surroundings, and it will enhance their interactions with the environment and other organisms. So this combination of sensory inputs allows the fish to thrive in lots of different aquatic habitats. They can use this lateral line system in order to avoid predators, or in order to find their prey, or in order to find mates. So again, all to help with the survival. So the evolution of sensory structures in amniotes. So the lateral line system, so this lateral line that these fishes and the amphibians have, disappeared in amniotes. So in reptiles, birds, and mammals, that lateral line doesn't exist. And this is because these animals are not living in water anymore. Okay, now the lateral line system is for basically basically providing information about water displacement in water, or water pressure. But there is no water in the air exactly, so there is no reason to have this lateral line system. It's not actually going to work. And the other thing is that in order for the lateral line system to be maintained, it basically needs water. So if we were to have this, it would lead to excessive water loss because our body would be constantly trying to provide water to this lateral line system, which isn't even helping us to begin with. Okay, so while the lateral line system has disappeared in amniotes, we still have, of course, some sensory mechanisms that will help us understand what is happening in our environment. So we have sensory cells called neuromasts also, and they basically have a new role within the inner air of the amniotes. So I don't know if you guys understand or know how hearing works, but actually, wait, we're going to cover hearing in the next syllabus point, so actually it's fine. But you have hair cells, okay, which are essentially going to be moved by the vibrations of particles which are going to go inside of the air. And when that happens, that will then eventually form a stimuli, which is going to go to the brain. And then that is how your brain is going to detect sound, essentially. So the neuromast, which have the sensory cells, also have the supporting cells, okay, with them, in order to support the hair cells basically around them. So the role of the hair cells in the inner air of the amniotes, the hair cells are integral to the cochlea. So the cochlea basically is a structure inside of the inner air that has liquid in it. And this liquid basically, it's going to be released and then it's going to push on these hair cells. Then when it pushes on these hair cells, some ion channels within the nerves are going to open, and then this is going to start that electrical signaling to the brain. And that is how we're going to determine that we've heard sound. The cells are also important within the vestibular system. So basically the vestibular system helps us with balancing. So the neuromasts that we're talking about here, which can be found in the amniotes, will basically help maintain balance and also will help to give spatial orientation. So, you kind of know which way you're facing and so on. Then there are the Merkel tactile discs. So Merkel tactile discs are important for fine touch perception in terrestrial vertebrates, which will allow the terrestrial vertebrates to understand differences between textures and shapes. So you can find the Merkel discs or Merkel tactile discs in the basal layer of the epidermis. It's at the base of the hair follicles and in hairless skin areas like the fingertips and the whisker pads. The Merkel cells, they basically have nerve endings, which will form the Merkel tactile discs. They act as mechanoreceptors that will respond to light touch and pressure, and then they will release neurotransmitters that will stimulate the nerve endings and then send signals to the brain. The yes, okay, so that's fine. The what's it called? The Merkel tactile discs. They function as slow adapting type 1 mechanoreceptors, and they're very good for detecting sustained touch and pressure, so just very, very, very light pressure, and enhanced texture and shape perception. So fine touch and texture discrimination. So the Merkel tact discs are important because they enable detailed discernment of object textures and surface patterns, which are important for activities that need this fine manual dexterity. So things like writing and typing is made possible for us because of these Merkel tactile discs that we have. Then the Merkel tactile discs have a high density within sensitive skin areas, so the fingertips, facial areas, facial regions, which will allow for high spatial resolution in touch perception. Then they also have developmental and adaptive significance. They enhance tactile feedback, which is important for various behaviors and motor functions, which will help us understand and explore the environment around us. And so basically in this way, we're able to use tools because of these Merkel tactile discs. Then we have the development of taste. So the development of taste basically has changed according to the dietary needs of the animals. So of course, all animals have different diets, and the taste has changed and adapted according to the different diets that we have. So the earliest vertebrates, they were aquatic organisms and they would basically use chemical cues to find food and to avoid predators, as we mentioned. So they developed chemoreceptors. And these chemoreceptors are what eventually would later become the taste systems in other animals. So the primitive chemoreceptors could detect a range of soluble compounds. Over time, these receptors evolved to become more specialized and then allowed organisms to detect distinct types of chemicals more effectively. This specialization included the differentiation of taste receptors that could identify basic categories like sweet, sour, salty, bitter, and umami, which is savory. Now, not every animal group is going to have all 5 types of receptors. All 5 types of taste receptors, and this is very highly dependent on what type of diet they will have. So as vertebrates moved from the aquatic to terrestrial environments, the gustatory system, which is the taste system, adapted to new challenges. Airborne chemicals are less readily detected than those in water. So basically, we then shifted reliance towards smell. But direct contact with the chemoreceptors is needed in order for taste to even work, really. So the taste buds had to then become developed, and they basically became more developed, more organized, and efficient. And these specialized structures basically have the taste receptor cells, and they can be found, of course, in the oral cavity. But in some species, like in some fish, they can be found on the external body parts as well, but that's only in some fish. Okay, so in mammals, the taste systems became highly complex because of the variety that we have in our diets and the complex social behaviors that we also express. Mammals have developed a refined ability to detect a range of tastes with high sensitivity, and this basically has been helped by us having a larger number and variety of taste receptors compared to other vertebrates. Then genetic variations and duplications have then helped in giving us more diverse taste receptors. So for example, the ability to taste sweet substances is highly developed in frugivorous animals. What are frugivorous animals? These are animals which have a diet consisting only of fruits. So if they're only eating fruits, their taste buds for, or their taste receptors for fruits, not for fruits, but for sweets, will be higher, okay? Because that is all that they are eating. All right. Now, the dietary specialization, as we said, yes, so the herbivores, the carnivores, and the omnivores have different taste receptor profiles, again, depending on the type of foods that they're going to eat. And then the ability to detect bitter tastes basically came from the fact that animals back then, of course, would not know what was toxic to them and what was not toxic to them. So as a general rule, the things that were toxic to the animals back then would taste bitter. Okay, so plants that were toxic would be plants that were toxic that had toxins in them would be tasting bitter. So after some time, these animals, when they first experienced these plants that were bitter, and of course a bitter taste is not the most pleasant of tastes, they would then quickly move on and develop a memory that, okay, cool, this plant that I ate tastes like this, so I'm going to avoid it. And this then, of course, also again helped with their survival. So they would be able to avoid these bitter or toxic plants, essentially. So the summary of taste reception. Okay, so chemoreceptors are basically what is going to help us detect the different types of tastes. We mentioned that we have taste receptors, which are specific to either which are specific to either sweet, sour, salty, bitter, or umami, like savory. Umami is associated with high protein foods. So animals which are constantly having a high protein diet, so for example, animals which are carnivores, are going to have better developed umami receptors because that is all that they are exposed to. Okay? There are some vegetables which would give a savory taste, but again, it's mostly meats that would have this umami savory taste. The umami and sweet taste would help the animals find nutrient-rich food, And then, of course, the bitter taste would allow them to avoid any food sources which would be toxic to them. The detection of salts is important for us to maintain electrolyte balance in the body. And the sour taste would allow the recognition of unripe fruits and spoiled foods. So whenever they would taste something, they would taste a food, and this food source would be sour to them, they would then again avoid it next time, because they'd be like, okay, something is wrong with this food. And then again, they are enhancing their lifespan this way. They're bettering their survival by avoiding spoiled foods which may be containing bacteria, etc. So primates, including humans, have a preference for mildly sour taste, especially when combined with sweetness, as it will allow them to look for foods which are containing vitamin C. Okay, so oranges, for example. The taste preferences and the sensitivities can vary based on their dietary habits and their metabolic needs, and this influences the food choices and the foraging behavior. So the foraging behavior is basically when they're looking for foods. So when they're looking for foods, they're going to look for things that have a mildly sour taste, but then kind of also have a sweet taste. So the taste buds are onion-shaped structures that have taste receptor cells. They are modified epithelial cells that have microvilli that come from the apical surface, which is at the top, to increase their contact area. Then they have supporting cells around them, which will maintain and renew the taste bud structures. And then the renewal process of the taste receptors is basically so that they are constantly constantly renewed by the division of the basal cells. So the basal cells would be in the stratum basale. As we said before, the stratum basale is the lowest not epithelial layer, but the lowest layer of the epidermis. Okay, so the taste receptor cells are constantly being replaced and renewed, because over time they may not be working as well. So that's why they keep getting replaced. Now, the taste papillae in mammals. So taste buds are primarily located on the tongue, and they have specialized structures called taste papillae. There are 3 types of taste receptor cells inside of the taste buds. We have type 1 cells, which will detect salty taste, type 2 cells, which have GPCR receptors, so a specific type of receptors, which will detect sweet, umami, and bitter tastes. And then we have type 3 cells, which will detect sour taste. In regards to remembering the different specific receptors for the specific tastes, you're not again expected to remember that. You're more so expected to remember how we're able to taste. So we're able to taste because of our chemoreceptors, and the fact that the chemoreceptors developed depending on the type of diets that these animals would eat. As we said, if they are frugivorous, where they're always only eating fruits, their taste buds for sweets is going to be more developed. If they're only eating meats, like carnivores, for example, they're only eating meats, their taste buds are going to be more developed for savory umami foods. Okay. Now, this is again a picture from the lecture slides. Again, this is not something that you are expected to remember, okay? But this is just here just in case you guys would want it. Okay, so the phylogeny of taste perception in fishes. In the evolutionary lineage of fishes, taste perception goes beyond the oral cavity, because as we said, some chemoreceptors in fishes can be found on the surface of their skin. Okay, so lampreys and fishes, their taste buds are not restricted to the mouth and pharynx, but can also be found on their body surface. This allows these fishes to sample chemical cues from their environment, and is especially helpful for animals which are living in murky, so basically very dark, very dirty waters, where they're not really able to see many things. So their chemical receptors, their chemoreceptors, are able to basically help them find, in a way, kind of help them smell, but not exactly, but they're able to find their prey in that way. So then the bottom-dwelling fishes, so species like catfish, they adapted to bottom-dwelling lifestyles, and because of this, they have whiskers that are covered with taste buds that will surround the mouth. These whiskers will serve as the tactile organs that will locate food and substrates, and it also gives them sensory input through taste perception. So in turbid waters, again, where the visibility is reduced and they're not really able to see things, these taste-equipped barbels or whiskers will allow the fish to be able to see, not see, but to be able to feel where they could find their next prey or food source. Now, the evolution of loss of taste in vertebrates. So over time, some vertebrates have lost certain taste buds. So the carnivores have lost the sweet receptors. And the reason for this is because they're not really eating sweet things. They're more so eating umami, meat-based diets. Because of this, they don't really need the sweet receptors because there's no sweet food that they're actually eating. Okay, so because of this, the sweet taste perception is less relevant for these animals because they're not really eating these sweets, or they're not really eating carbohydrates, which would, because the carbohydrates, they would become glucose, and this glucose would then be classified as a sweet by the taste buds. The herbivores, which are only strictly eating plants, would lose the umami receptors. So specifically pandas. Pandas, they don't have that umami receptor. They're constantly eating their bamboo trees. And when they eat these bamboos, the bamboo itself doesn't have umami-rich components. Because of this, pandas, they don't have that umami receptor. And this basically aligns with their dietary focus on only eating plant material instead of eating protein-rich foods. Because remember, we said umami receptors are able to detect the umami flavor because the umami flavor is rich in proteins. But if you're not eating proteins, then these animals would not have the umami receptors. Now, the umami receptor loss in bats. So all bats, regardless of their dietary preferences, don't have umami receptors. This can be seen specifically in vampire bats. So these vampire bats, they only drink or eat blood, okay? And blood is not a significant source of umami compounds. It doesn't really have a lot of protein rich parts to it. And because of this, and because they only eat blood, that, well, there was no need for them to have the umami receptor, so eventually they lost it. They also lost the sweet receptor for this very same reason, because these bats are not eating carbohydrates. They're literally only feeding on blood, and blood doesn't have sugar. And because of that, the taste buds are not, they're not being utilized. So if something is not being utilized, eventually that structure is going to go. Humans, as omnivores, we eat lots of different foods. We don't just eat meats, we also eat plants. And because of this, we have kept all our taste profiles. We've kept our sweet, our sour, our salty, our bitter, and our umami. Okay, and this all is as a result of the dietary preference that we have, or the foods that we eat. The olfactory organ, I think, is the last one. Yes, the olfactory one is the last one. So the olfactory organ is responsible for the sense of smell. And it basically consists of the olfactory epithelium, which lines the nasal passages. Okay, so the olfaction has a critical role in detecting volatile or water-soluble substances, which are called odorants. And this could include food components, which could indicate to them danger, or pheromones, so that they're able to basically find a mate. So the embryonic formation. In embryonic development, the olfactory organ comes from a thickening of the surface ectoderm called the olfactory placode. And this thickening happens because of the neural crest cells. The neural crest cells, remember, we said that they are important for embryonic induction, as they basically tell the cells around them what to become. So the neural crest cells essentially tell the ectoderm to form the olfactory placode, for the ectoderm to become thicker and form the olfactory placode. These placodes are then going to invaginate, so they're going to form pits, basically called nasal pits, and these nasal pits are going to develop even more and become nasal sacs and passages. Now, the cell types that are found in the olfactory epithelium, you have three. You have the olfactory receptor cells, the supporting cells, and the basal cells. The olfactory receptor cells basically are neurons that have dendrites that carry cilia or microvilli to help the olfactory receptors. And then the supporting cells will secrete mucus to cover. The supporting cells will secrete mucus to cover the olfactory epithelium and to maintain moisture. And we need this moisture again for the olfactory, so basically for the sense of smell to be working at its maximum capacity, not capacity, but at its level best. This moisture is needed. The basal cells then have stem cells. They're stem cells that renew the olfactory epithelium, including the olfactory receptor neurons. So again, as we said, the cells and these neurons need to keep getting replaced so that the olfactory can be improved time and time again. The olfactory receptor neurons have a short lifespan and are continually replaced by differentiating basal cells. We said that these basal cells can be found in the stratum basale, which is the last layer of the epithelium. So the olfactory epithelium is a site of adult neurogenesis. Adult neurogenesis basically meaning the formation of nerves whilst in the adult stage. The importance of olfaction we've already mentioned. So the olfaction in phylogeny. So early vertebrates, the olfactory organ was unpaired and connected to the pituitary gland, and this basically stopped jaws from being formed. And that's why they didn't have jaws. That's why the Gnata didn't have jaws. But in gnastostomes, which are the animals which have jaws, the olfactory apparatus became paired, and this allowed these cartilaginous fishes to be able to have smell direction. So basically, they're able to detect smell and then follow the area of where that smell is coming from. Fishes, except for lungfish, have isolated nasal passages that are dedicated only for the sense of smell. The nasal sac is elongated and it usually has an olfactory rosette structure, which you can see right here. It kind of resembles a rose. And the water flows through the anterior. It flows through the anterior nostril and then goes to the posterior nostril. So we have the anterior and the posterior. So this would be the anterior and then the posterior. So the anterior would be here and then the posterior would be at the bottom, or vice versa. Then the olfactory organ as a portal for pathogens. So olfactory receptors, neurons, they provide a direct pathway from the external environment directly into the brain and therefore bypasses the central nervous system barriers. This can make the olfactory nerve vulnerable to infection by different pathogens because they have a very easy way in. Okay. Now, the olfaction in terrestrial vertebrates. So terrestrial vertebrates, specifically tetrapods and amniotes, have gone through significant adaptations to accommodate transition from the aquatic to terrestrial environments. These adaptations include changes in their nasal anatomy and their development of specialized olfactory structures and the differences and whether they actually have a vomeronasal organ or not. Okay, so the migration of nostrils would be the first thing that we'd need to discuss. So remember how we said that the fish have the anterior nostrils and the posterior nostrils, right? So the posterior nostrils actually in amphibians would move into the oral cavity of the amphibians. So the posterior nostrils, so the ones at the back, so number 2 here, the posterior nostrils are going to move into the oral cavity when they become amphibians. And this allows for breathing through the nose and transforms the nasal passages into upper airways. So before this, the fish that only had the anterior and posterior nostrils only on the surface, like right here, rather than inside the mouth, these fishes would only have their olfaction organ only as an organ for smell, rather than also as an organ for breathing or respiration. The transition to air increases the importance of mucus in the olfactory system, as it helps to trap odorants and facilitates the detection by the olfactory epithelium. So in amniotes, the dorsal part, so the back part of the nasal cavity, is important for olfaction, and it has been widened and then has formed skeletal protrusions called the olfactory conchae or the turbinates. These structures then give additional surface area for olfactory epithelium, which will again improve the sense of smell. So mammals, including primates like the humans, they have lots of different sizes and complexity of the olfactory conchae. And primates generally have diminished olfaction compared to other mammals. So basically, compared to other mammals, we have reduced sense of smell. So for example, cats and dogs have a much better sense of smell than we do. The vomeronasal, or the Jacobson's organ, now this organ is something that most tetrapods will have, and this detects odorants with low volatility at close distances. So volatility is basically the gases. Basically, it's how dense the gas would be. So basically, the vomeronasal improves the olfactory. So it basically allows them to smell things which they would have otherwise not been able to smell had they not had this organ. So they can also smell things like pheromones in this way. The vomeronasal organ can be found in the vault of the oral cavity at the base of the nasal cavity, and it connects the oral cavity through a duct called the nasopalatine duct. And then the defects of the vomeronasal the defects of the vomeronasal origin. So the vomeronasal organ is underdeveloped or entirely lost in certain groups, including secondarily aquatic, flying, and some arboreal species. So basically, species which so they don't live their whole life in water. They live some part of their life in water, and then species which will be flying also. They may have lost this vomeronasal organ, meaning that their sense of smell will then become poorer. So then we have the anomalies within humans. So the nasopalatine duct, basically, it becomes removed in fetal development in some cases, and then this can lead to some anomalies, so some illnesses, not illnesses, but some defects, where basically these what's it called? These humans then that have these oh my god, these humans which have these anomalies, right, where the vomeronasal organ is not developed properly, will not be able to detect these smells, right, or these pheromones. Okay, so here we can see this is where the olfactory epithelium is. Okay, it covers the part of the nasal cavity. And we said that it has mucus in order to really trap the odorants, okay? In order to trap the odorants so that we're able to detect these odorants. The vomeronasal organ is right here. In humans, it is quite small because we don't really have this ability. I mean, we have the ability to detect odorants that are in much smaller concentration, but in comparison to other animals, like as we said, dogs and cats, they have a better defined vomeronasal organ than we do. Okay, and that was the end. Okay, does anybody have any questions? Or does anybody want me to explain something again? No? Okay. We are at the last slide now, which is about the phylogeny and the development of the ears. So the ears are responsible for two things, of course, for hearing and also for balance. So the ear is also known as the oris, and it has three main parts. So we have the outer ear, which is the part that you can touch. It's the visible external ear canal. And then we have the middle ear, which is in between the outer and the inner ear, and this is usually the part that has the eardrum. It also contains small bones, and it connects the tympanic membrane to the inner ear. Then we have the innermost area, which is the inner ear, and this basically houses crucial sensory structures that are important for hearing and balance. It includes the cochlea for hearing and the vestibular system for balance. So the phylogeny of the ear. So the agnata or the cyclostomes, they had limited inner ear structures. So the lampreys and the hagfishes, they have very simple inner ear structures with a single cavity and a few semicircular canals. And this basically means that they're swimming. This is basically because of their swimming. Because remember we said that the ears are also important for that balance. They don't really have very well balance. They don't really have very good swimming abilities. And because of this, their semicircular canals would be less developed. Then the cartilaginous or bony fishes have well-developed vestibular apparatus. They have 3 semicircular canals, a utricle and a saccule, and this helps their balance. As they become better swimmers, they need to have this balance, whereas the agnatas, they don't need to have a balance specifically because they're not very good swimmers to begin with. Now the amphibians, like frogs and toads, so the anorans, like frogs and toads, they basically develop the middle ear, and you can actually see their middle ear on their head. Now this middle ear is there because it has the eardrum, and it's important for sound amplification. So basically, the amphibians moved from living in water to living on land, right? And water is not really the best conductor of sound, whereas air is. So for that reason, they had to develop the middle ear so that they'd be able to hear things. Now they'd be able to hear things basically much better because of the presence of the air. So the higher mandibular bone that we spoke about yesterday moved. So it was found first within the fish, kind of close to where the gills were. So the higher mandibular basically moved from where the gills were, and basically moved closer to where the middle ear was going to develop, as it would be there to help to transmit sounds to the inner ear. And then here is the structure of the middle ear. As you can see, consists of a eardrum and a stapes. Then here we have the inner ear, which consists of the saccule and the utricle, which contribute to the balance, as we said, of the cartilaginous fishes. Okay. All right. So reptiles and birds have improvements in their hearing because of the elongation and settling of their basilar papillae. Now, the basilar papillae is similar to another organ called the organ of Corti. Now, if we look at this picture, we will be able to see that the basilar papillae, or the organ of Corti, is this purple thing. So what we're saying is that the basilar papillae essentially became elongated, as you can see, in birds and crocodiles, and this again enhanced their sense of hearing. This basically meant that they'd be able to hear sounds at different frequencies, so at a different range of frequencies. So the more developed the basilar papillae, the better the frequency of sounds that you'll be able to hear. Now, some reptiles, they don't have a distinct outer ear, whereas crocodiles and birds have very well developed ear canals. So the ear canal, not the ear canal, but like the outer ear, it basically makes it so that sound is perceived much better, because the air, the whole point of sound is to vibrate. So it would be basically vibrating air molecules, right? These vibrating air molecules would hit your outer ear first, and your outer ear would basically collect all these vibrating air molecules and would basically guide them into the ear canal. So we said that the reptiles, so the crocodiles and the birds, they don't have a distinct outer ear, but they have a well-developed ear canal on the inside, within the middle ear, which is going to help them with this function. So In the evolutionary history of vertebrates, the outer ear has gone through significant changes, specifically in the reptiles and mammals. So in the reptiles, there's been tympanic membrane placement. So in many reptiles like iguanas, the tympanic membrane, so the the eardrum, is found at the surface of the head, which is similar to the frogs and the toads. And because of this, because their eardrum is right there, they don't really have a distinct outer ear structure. Then some reptiles, like skinks and agamids, so basically bearded dragons is an example, the tympanic membrane can be found at the end of the short ear canal, and crocodiles and birds have longer ear canals to help with their sound transmission. Okay, so mammals basically formed they formed a significant elongation, okay, which basically would spiral to fit part of the temporal bone. So this spiral would be the cochlea, and we can show you here. So as we can see here, the cochlear duct essentially, basically, as we can see, so at first, the legana is basically what's going to become the cochlear duct. So we can see in the legana of the aquatic tetrapods, the amphibians, the lizards and snakes, the legana is not very long, right? But in mammals and birds and crocodiles, the legana actually first of all becomes the cochlea, and the cochlea basically spirals in mammals to fit into the temporal bone. Okay. Then the ancient sensory structure, which was known as the macalo-laginae, which is found in the laginae of non-mammal vertebrates, is lost in mammals, but recent studies have shown that this structure could have a role in the bird's ability to sense the Earth's magnetic field. So that's just a quick fact. I mean, if you mention this in the exam, they would think that you've read the lecture slide, so I would say try to remember it. So the mammals, they have 3 auditory ossicles. They have the malleus, the incus, and the stapes, and all 3 of them help to transmit sound vibrations coming from the tympanic membrane into the inner ear. As we said, the auricle or the pinna is the outer ear of mammals. So some mammals would have this. It's a characteristic feature usually of mammals. Not all animal groups will have this pinna or the outer ear, as we mentioned. We mentioned that the pinna collects and directs the sound waves to the eardrum, into the canal, into the tympanic membrane. Unlike humans, most mammals have well-developed muscles that are attached to their pinna, that is attached to their outer ear. This allows them to move and rotate their outer ear so that they're able to capture sounds from different directions. A good example, again, of animals that do this are cats. So cats basically are able to rotate their ears in the direction of sound in order to better hear what is going on, basically. And I'm sure that a lot of you have seen this before. So the organ of Corti is a structure found inside of the cochlea of the inner ear, and is important for hearing invertebrates, including in humans. It has an important role in auditory transduction, and it converts sound waves into electrical signals. So the organ of Corti is found on the basilar membrane inside of the cochlear duct of the inner ear. It is made out of sensory hair cells. So for example, the stereocilia. So stereocilia are a type of hair cells which you can find there, and you can find other structures which are arranged in complex organization. So there are 2 types of sensory hair cells, the inner hair cells and the outer hair cells. These hair cells are mechanoreceptors that will detect sound vibrations. So the stereocilia are hair-like projections, and they're arranged in rows kind of resembling stairs. These stereocilia are deflected by the movement. So whenever the sound waves come and enter the air, and specifically come into the inner ear, the sound waves are going to kind of bend the stereocilia a little bit, okay? Above the hair cells, there is the tectorial membrane, which is a gel-like structure that will cover the hair cell stereocilia. When the sound waves basically bend the stereocilia, what this will do is it's going to release a liquid. So the cochlea is going to release a liquid, and essentially this is then going to start it's basically going to influence the nerve cells to open up some ion channels to allow an influx of ions to come in. When this happens, a nervous stimuli is then formed and then, of course, sent to the brain. So when the stereocellular bend, it opens mechanically gated ion channels and then allows these ions to go into the hair cells. This generates the electrical signal, which is then transmitted by the auditory nerve to the brain so that the brain can interpret it. So the organ of Corti is important for converting mechanical sound waves into electrical signals that can be understood by the brain. It's very important for auditory processing. It allows us to perceive sounds of different frequencies and amplitudes. And if there's any damage or dysfunction, it can lead to, of course, hearing loss or hearing impairment. So the organ of Corti and the cochlea are very, very important. So just remember that the organ of Corti can be found within the cochlea. Okay, so now the development. So as with all sensory organs, we first are going to start with a placode. So the otic placode is going to form first. So the air development starts off with the formation of paired otic placodes on either side of the embryonic head. They're going to be adjacent to what is going to be the rhomboencephalon later on. These placodes are then thickened areas of surface ectoderm. So here we have the otic placodes that we're talking about. So we have the otic placodes basically on either sides. These are the otic placodes. Okay. Now they're going to form the otic vesicles. The otic placodes are going to invaginate to form the otic pits, which then are going to detach from the surface to become the otic vesicles, which are the fluid-filled sacs. So here is the otic pit, right here. Okay, so it's going to separate itself right there and become its own kind of, it's going to have its own membrane, we could say technically, like that. So essentially, we have the otic placode right here, which is the ectoderm. The ectoderm is going to thicken up. So it thickens up and then eventually it forms a little bit of a pit. As you can see, here is the invagination. It's forming the pit. It forms the pit and then it kind of cuts itself off right there, and then it forms the otic vesicles, which are carrying, they're basically fluid-filled sacs. So next to each vesicle, cells from the placode and the neural crests assemble to form a ganglion. This ganglion is going to basically help with the nervous impulse later, which we need. This ganglion is going to split into the vestibular and the spiral ganglions, so the cochlear ganglions. And these are then going to respectfully basically make or contribute to the parts of the air which are responsible for balance and the parts of the air which are responsible for hearing. Okay. Any questions? Because we're at the close to the end, actually. I thought we were at the end, sorry. Any questions so far, actually? No? Okay. Now the development of the membranous labyrinth. So the utricle and the saccule, which are structures which are found in the inner ear, the otic vesicle basically is going to give rise to these two membranous sacs, which are the utricle and the saccule. They're going to form the membranous labyrinth of the inner ear, as we mentioned. From the utricle, 3 semicircular ducts are going to form in perpendicular planes. So they're going to be perpendicular to each other. We're going to have the anterior, the posterior, and the horizontal or lateral ones, and each will have a extension to it, which is going to be called the ampulla. In mammals, the saccule will form a helically curved outgrowth, which is called the cochlear duct, as we mentioned, which is important for hearing. As the otic vesicle will sink, the surrounding mesenchyme is going to form the cartilaginous otic capsule, which is going to later ossify. So it's going to go from being a cartilage to bone. So it's going to become bone and it's going to form the bony labyrinth. So the bony labyrinth is formed from the mesenchyme. The otic vesicle comes from the ectoderm, right? And the otic vesicle will give rise to the 2 membranous sacs that we mentioned, so the saccule and the utricle. We said the utricle is going to make the circular ducts, okay? And that is enough to mention. Oh, sorry. Yes. Okay. So now the fluid composition. So the membranous labyrinth has got endolymph inside of it, while the bony labyrinth has got the perilymph inside of it. So here we can see everything that is blue here, right? So including the cochlea, is the bony labyrinth. And as we said, the bony labyrinth has perilymph in it. The membranous labyrinth is right here on the inside, okay? And the membranous labyrinth, essentially, we said it has endolymph. These fluids have different ionic compositions, as in the endolymph has more potassium ions, which is important for mechanotransduction